Our next speaker is Dr. Kristen Marhaver with Karmabi in Curacao. And before we bring Kristen up, I want to tell you two of the reasons that I absolutely love Kristen as a friend and a colleague. The first is when we were doing our meeting 2 years ago, the workshop, she was the first one to shoot her hand up when we said, hey, we've got an idea. We want to do maybe this webinar series every couple months. And she said, I'll do one. But the best part of that was when she pulled me aside and asked me if it was okay if she swore it on the webinar, because that's just Kristen, for those of you that know her. The next thing is because she was willing to jump on board as part of one of my many crazy ideas a few months ago. Those of you that know me well know I'm a big fan of the stand-up to cancer approach to research. For those of you that aren't familiar with that, they basically say we're going to put aside our academic pursuits and we're going to form dream teams, the best of the best in individual fields. Not everybody has to know everything about the whole problem. We want to bring the best team together. And so a few months ago, actually just last May, not that long ago at all in the big scheme of things, I was having a conversation with our colleagues at the Paul G. Allen Family Foundation in Vulcan about the idea of using assisted gene flow as a tool for restoration. And they said they loved that idea. They were thinking the same thing, but didn't think anybody was really ready to operationalize that we were still a few years from figuring out how to make that happen. And I said, I disagree. I think actually we can make this happen, and I think maybe there's even a chance we can do it this year. I think all the pieces are in place. We just don't have the team to execute it. And so I told them if they found the money, we would find the people. I, of course, didn't have any people committed at this point, and knew that my first calls needed to be to two people that would be key to making this happen. And the first was Dr. Mary Hagedorn with the Smithsonian Institution, and the second was to Kristen. And while they totally told me no when I first asked, I didn't take no for an answer. And they told me I was crazy to think that we were going to execute a project to, for the first time ever, do cryopreservation and translocation of genes using assisted gene flow. And I still pushed harder, and after much cajoling, I finally got Kristen and Mary on board with the concept. After that, we knew that while they were the experts in cryopreservation and in larval rearing, that we would be nowhere if we didn't have a team that could take care of those babies if by small chance we succeeded in this for the first time ever. And so with that, we needed to bring Carrie O'Neill on board with the Florida Aquarium and Chris Page with Mote Marine Lab, because they are by far some of the two experts in this side of the world, at least, in rearing up little baby corals. And so with that, we had our dream team. And Kristen's going to tell you a little bit about that project and what happened there, and also some of her other amazing work that she's doing and her lab is doing that's going to make that project look like child's play. So enjoy Kristen for the next 30 minutes, and then we'll have 10 minutes for questions when Kristen is done. Thank you.
Thank you very much, Tom. I like that your number one memory is me requiring permission to swear. I recently did a thing for little kids, and I managed to not swear on like a live stream for 45 minutes, and that was a big achievement. And I was like, okay, I'm done. I proved I could do it once. I never have to do that again. So that's cool. So the team invited me to talk about larval propagation and next generation technologies, but the only way you can understand where we got in the world of larval propagation is by reflecting back on how much improbable progress we've made. And so this talk is just as much, I sort of changed the title of the talk, and I challenged myself to go back and think about how we got here, and then try to maybe give some ideas or some advice about where we could go next. So first of all, this is a tricky audience because there's the world experts and also people who have no idea what the larvae people are up to, but just think we're all nuts. So how many of you have worked on coral larvae before? Let's do a show of hands. Okay, that's pretty good. How many of you have never worked on coral larvae before? Yeah, okay. How many of you worked on coral larvae but stopped because it was too much of a nightmare? Don't be shy, don't be shy. Yeah, they're all in a group for some reason. The larval recovery support group is back here in the audience. How many of you would work on larvae or are interested in working on coral larvae but don't really know how, but if you had a way in, you would be interested to start? Cool, okay, that's awesome. Coral larvae research has been extremely difficult for an extremely long time, and when I first started working on coral spawning, it was totally normal for a field season to end with nothing to show for it. So in Panama in 2005, I tried to do an experiment for Niggy Fogarty with all these different crosses between these different species of what was called Montastria at the time, and nothing fertilized. And then it was like, I guess we'll try it next year. So really, my momentum in larval propagation started when I started working at Carmabi. Mark Vermey and I showed up in 2007. The whole place was kind of ramshackle and run down. Ayanna Johnson was our field assistant, who later found out she had a much brighter future ahead of her that had nothing to do with larval propagation. And the three of us sat there and figured out how to raise larvae in the little tiny yellow building on the left-hand side there. And the reason that it was sort of a founding moment, and it was useful to start from scratch with no traditions in place, because Mark brought over the extreme paranoia he had been using in Hawaii, working on Montipora capitata, and we then applied it to a really happy, healthy reef system that was making tons of babies and was giving us really good material to work with. So just 5 minutes down the street from Carmabi, there's reefs that still look like this, with pretty decent herbivore populations, and there's a little dendrogyra hanging out back in the back there. And so the progress that we've made is because of all the people who worked so hard to get us any kind of footing, and then these corals that have done such a good job giving us nice stuff to work with. So this is my favorite view of Curacao, but there's also the view of the larvae lab where a lot of the magic happens. There's some new labs there that are a little bit fancier, and then if you've ever been there, you know mostly what you see is us sitting around drinking beers at tables. Not a lie. We just finished 4 months of coral spawning. We started in July. My last coral spawning dive was November 18th, and the only reason we stopped is because Thanksgiving happened. And so we just spent 4 months at picnic tables drinking beer, trying to understand the shape of reality and where we fit in it. And it feels like no matter how tired you are, you're always drawn back to that picnic table. The beer tastes better when you've worked harder, and there's always more stories to tell and more anecdotes to share. That's where we come up with the ideas, that somebody runs out the lab and says they're dividing, and somebody runs into the lab and says, I've got an idea. And this fizz and this foment has really been the secret to us making the progress we have. While we're on the topic of beers, if you have the number 84 on the back of your badge, I would like to meet you back here at the end of this session. Thank you very much. Just want to make sure I didn't forget that. So I'm going to tell you guys a lot of anecdotes today. And it's partly because every time we sit at this picnic table talking about coral spawning and how we just caught something or saw something we'd never seen before, I get this sort of like tingly feeling in my skin. And the only way I can understand how change happens and how space and time work is this cheesy quote from Bill Gates. We always underestimate the change that'll occur in the next 2 years, or overestimate the change that'll occur in the next 2 years, and underestimate the change that'll occur in the next 10. Then he says, don't be lulled into inaction, which I think doesn't apply to this audience, so I blacked that part out. Just like it's not an issue here. This quote, I think of this quote all the time, because in a 2-year time scale, maybe nothing really changes, but if you look at where we were in coral propagation 10 years ago, it's crazy where we are now. So the quick outline for this is I'm going to talk about 3 big milestones from the past of larval propagation, 3 breakthroughs that we're working on right now in the present, and then 3 unpopular opinions about where we go in the future. Just keep some dramatic tension. Okay, the first milestone is that this works. What? Let's just stop for a second and appreciate how totally insane it is that any of this works at all. This Favulata colony does this for 1 second per year, and we know when it happens and where to find it. That's Mark not kicking the coral, but he kicks me right in the few seconds in this video. This was maybe one of my favorite ones that I ever watched. It's crazy that we know when this happens. Somebody had to invent the aqua lung and be scuba diving in the middle of the dark night to figure this out. It's crazy that we know that this happens. It's crazy that this happens. It's crazy that that works. There's a Cropera palmata this summer doing what they do. It's crazy that that works, and it's crazy that we can foster them through this process of embryogenesis and see it happen before our very eyes. And it's crazy that we can then see these little tiny guys swimming around trying to figure out where to live for the rest of their lives. This didn't always work. The early days of larval propagation and coral spawning were tough. It was so hard to keep them alive that the objective was always a big scale. Get as much as you can because most of it's going to die. And then for no explainable reason, everything would just crash. They would get overgrown by this hot, pink, crazy bacteria. Everything would just die. You'd go in the next morning, it was all dead. There's like a weird echo. Can you guys hear the feedback? Or is it just me up here in this little, there's just a little bit of feedback. This didn't always work. And that made for a pretty rough study system. There were only a couple species in the Caribbean, not that many chances per year. You're always doing it at a huge scale. Things were always going wrong, and a lot of people were screaming and crying and yelling, and people would swear off coral spawning for the rest of their careers, which it makes perfect sense to me, truly. It's also one of the meanest games you can play, because every step you have to get perfect before you can even try the next step, but you only, you have to do every step perfectly before you can try the next one. And so it's like literally a sudden death game with an endangered species, and you only get to practice it one time a year. So anybody who stayed involved in larval propagation was high on a kind of euphoria that like doctors should study, because this was a super crazy thing to do. This was really nuts. But like, I just get this one chance, and if I get a little bit further, then maybe next year I can get a little bit further. Literally a sudden death game. We were sitting in the picnic tables at like 5 a.m. this summer, and the embryos were dividing in the lab, and I was drinking a beer with Mark, and I looked at him and I said, if we weren't really good at this part, I would be shitting myself right now. Those are my exact words. This is the hardest part. This is the part where everybody gets the most panicked, or had for decades, getting embryos to divide and successfully turn into larvae without the whole thing crashing. And there was this huge project with a ton of pressure riding on us, and a ton of anxiety, and I said, this is supposed to be the scariest, worst, hardest part, and I know it's gonna work. Why? Why do we know? How did we figure this out? The more aggressively paranoid we got about everything, the better we got. Super clean water, super clean plastic, super clean hands, clean karma. To ask me about that later if you want to know, but we have like a whole karmic like rejuvenation system at Karmabi to make sure we're not pillaging the reefs and then having like the forces of the universe bring revenge back upon us. Just being super paranoid about everything, keeping things as absolutely the lowest density you possibly can. One little baby should only be in 1 milliliter of seawater or more. And we found that the fewer we tried to keep, the better they all did. If we get super clean and then be super humble and ungreedy about what we take, it all goes super well. So somebody was saying, well, we need this many embryos because half of them are going to die, and Val Chamberlain and I looked at each other like, no, they're all going to survive. We don't know what that part is where they die halfway through. If they divide, we'll make it happen. This is basically what engineers and in manufacturing they call a 6 Sigma approach. So you can make a bunch of widgets and be happy if 10% of your widgets are garbage that you can't sell. Or you can say, I'm going to make my manufacturing process so strict and rigorous and so controlled that my widget has to get 6 standard deviations away from the average widget before it's a failed widget. So if you get 6 standard deviations away from your average output, you're talking about 3 widgets per million widgets. And then there's all this like management philosophy and a philosophy of change about how you find the places in your manufacturing process where you can smoosh your standard deviation and buy yourself that cushion. That's kind of what we did with larvae. We got so nuts about so many steps in the process that now we have an extremely predictable system. If you do the system, you still have room for like 10 things to go wrong, and you're like, they're still gonna survive. My students one day, the other night, were like, what is the goal of this dive? And I said, the goal of this dive is everybody survives. which then became sort of like our lab theme. So now it's like, what's the goal of this experiment? Everyone survives. And the goal of larval propagation now is everyone survives. Let's be ambitious, right? Those are all pretty vague descriptions of what we figured out in larval propagation. But if you would like to know all the gory details, we made this webinar for the Coral Restoration Consortium. With the amazing Val Chamberlain and Nikki Fogarty, we assembled basically everything that we know about the technical details of raising larvae. And you can listen to 3 hours of us talking about it, or you can download 140 slides about it at this website. It's always cool when your slide is just a picture of other slides. And that brings us to the Coral Restoration Consortium. Can we please give them one more round of applause? Holy shit, guys. This was not a foregone conclusion. There was a bunch of ambitious people who made this restoration real, and the magic that they have set in motion gives me chills and goosebumps as well. In particular, the opportunity to do that webinar was great because we had never kind of come up above water to collect all of our thoughts like that before. And then the chance to have Tom and Tolly connect me with Carrie O'Neill and Chris Page and Mary Hagedorn and Katie Lohr and do a collaboration together this year has been so powerful and so moving. I feel incredibly humbled just at the chance to be exposed to these masters at the helms of their ships. And that's all thanks to you guys making this into a reality. Our milestone 3 is that this actually works for a lot of species. It used to just be a couple species in the Caribbean that you could work on. So even if you did really well, it was only 2-month studies, a 2-month field season, and only 2 species to work with. But now we're like, let's figure out Psittacea spawning, and let's figure out Stephanocenia spawning. And Margaret Miller pushed us to go figure out Dendrogyra spawning, and we made some progress on that one, and they were super cute. They were super cute. The more that we've been able to make our system at Carmabi predictable and consistent and rigorous, the more fun we get to have. So we know those larvae are going to be fine. Let's go dive for crazy stuff. Let's go try to find a Dendrogyra spawning the day before coral spawning really starts. And so there's an important kind of trade-off there where if you can be calm about your core operations, you get to go have fun and tinker in the next sort of puzzle over. A couple of years ago, Mark and I, after figuring out Dendrogyra, were like, let's do all the Meandrinidae, excuse me. Let's figure out all the other weird ones too. Oh, I'm sorry, I forgot about the slide. So here's a bunch of the species we can raise at Carmabi, and there's even more that wouldn't fit on the slide. If you know something about raising a Caribbean coral species from larva to settler, tell Valerie Chamberlain, because she's making a guide to each species and their quirks so that you can get good at one species and then you'll have the exact information you need to know to transfer it over. Tell Val everything you know. Thank you. Or tell me and I'll tell Val. So we made a bunch of progress on a bunch of species, but this one was elusive for a super long time. How many of you ever seen Dickocenia spawn? 1. Find me later. Oh. 1. Yes. Okay, find me later. Are you Lori? Yes. Okay, we had 1 observation. I can only see a hand and not a head. We had 1 observation. There was 1 male Dickocenia spawned in Florida this night a few years ago. And Mark and I were like, how hard could it be to figure this one out? So we went diving and saw it spit out an egg-sperm bundle, which turned out to be eggs from Colpophilia. Ellen Mueller and Bonaire saw them spit out eggs that were yellow. It turned out those were worm eggs from a worm it had eaten. At some point, we're like, we don't want to see it spit out eggs. We want to see it spit out its own eggs, ones it made itself. No cheating. It was cheating. And then what happened was Mark was on a dive right before Favulata spawning when nobody dives, and found this. Ready, go. And that's why we didn't know when it spawned. That's the whole thing. But once we had that pin, we're just like, oh, it's no wonder we missed it. It was pretty subtle. Once we had that pin, then we knew exactly when to dive. And then we went night after night after night, and we dove 9 nights in a row, and we saw these things spawn all 9 nights. And we're like, now we don't understand coral spawning anymore. We don't understand how this works. But then we knew the block of time to watch, and that's like, all right, we're going to solve this puzzle. So you got to find the males. You got to find them spawn. They do it for about 2 seconds, and they're also light sensitive, so you put your light on it, and they spawn, and then they stop. So you have to know from far away, and then you have to like sneak up and shine your light and try to catch the sperm really fast. And so we're like running around this reef with syringes trying to catch sperm, but then you have to find the females. Like, where are the females? It took us, I think I saw 1 last year spray all its eggs, and then they were gone. And I think we caught like 10 eggs, and one of them turned into a 4-cell embryo, and then it blew up. So this year, we definitely didn't have a video of that. We had a picture of like 5 eggs coming out of a female. This year I found one, I was like, oh, I can tell that's actually spawning. So I have this very Sasquatchy video, but it does show what's happening. There we go. Yes. Yes, I could do that one more time. There's like filmmakers in the room like, what is wrong with your buoyancy control? There's a little bit of a prettier picture of a female spawning. So 15 months ago, we had never seen it. Lori had seen one. We saw one on accident. We started following them, and they wouldn't stop spawning, but we couldn't figure out how to raise them. And then this year, after 3.5 months of crazy coral spawning, Mark and I were just like, let's do this. Let's fucking figure this one out. So there you go. That was what you didn't know on that title slide is that you were looking at the embryogenesis of Dickocenia, a coral that was proposed for the endangered species list, but not listed on it, but still one that we've basically known nothing about until now. There you go. Ding. That has been one of the funnest parts about being at Karmabi, the opportunity to go take a puzzle and solve it. Just say, let's just keep diving and let's keep figuring this out. Oh, there we go. And so I think maybe unless somebody has a secret skunk works that works on Dickocenia that I don't know about, I think this is the first little Dickocenia settlers that we've ever seen as a group. And so that brings us to settlement, and that brings us to emerging breakthroughs. We got pretty good at larval propagation, and we're pretty consistent about it. And then once you get to the settlement stage, it's like, what are the rules of this stage? We've finally gotten to where we can be more aggressive about figuring out the rules here. So 3 emerging breakthroughs in larval propagation. The first one is getting robust settlement to happen. For a long time, it was relatively easy to get the larvae, and then you had no idea what they wanted to stick on, or they all settled and they all died. There's some pretty cool work happening that, like Chris Page and Carrie O'Neill, you guys are geniuses at settlement. Val Chamberlain has made all sorts of crazy progress in this. Anya and Unam has done a good job of figuring out how to get settlers to grow. Everywhere, all around the Caribbean, people are slowly hacking together what works at this stage. And as you can see from this photo from Chris, it's a pretty clean operation as well. For a long time, we thought you needed these crazy, complicated CCA communities and these really well-developed fouled surfaces, but you just give him a tank and a clean substrate and a little bit of CCA and a pretty low density of larvae, and he gets some pretty amazing magic to happen just in that controlled environment. So I think we're starting to see, actually Val has seen some of this as well, where if you have lots of CCAs in a surface, sure, more coral settle, but then they all get plowed over by the fast-growing CCA. So I think we're starting to see that being really clean and controlled about stuff is also maybe a better way to play the game at this stage as well. What we want to understand, though, is not just what to use and the recipe for what to use, but why corals settle and how we can trick them into settling where we want. So we're doing a new project at Karmabi that's a collaboration with University of Illinois, Urbana-Champaign, and San Diego State. And we're bringing the material scientists and the fluid dynamics physicists and the microbiologists, and we're going to study the physical and chemical and structural factors that make a coral stick, and the fluid regimes that go from laminar to turbulent that provoke corals to explore and that convince them to settle. That way, when we know the factors that are triggering them, not just the things, but the factors, we can go say, let's make a material that has all those factors, that does that to ocean water, that triggers those physical interactions that corals want to have with a surface. I'm super excited about this because I feel like I have the easiest job in the whole thing. I'm just like, here's some larvae, do the hard part now. Okay, emerging breakthrough 2 is getting these little dots to survive. This is an Acropora palmata from the Sea Corps program, and this is one of my favorite stories for a couple of reasons. Let's see, the Sea Corps started working in Curacao a few years ago, and then Val Chamberlain came aboard, I think, right after they had raised this group of Acropora. So she was in charge of outplanting the year-old palmatas. And at 4 years old, they started to spawn, and they're kind of on like a shallow seawall, so they were never collecting from these during spawning before. But they were, I think they showed for the first time in the Caribbean that an outplanted settler can then grow and reach reproductive maturity, which nobody knew for sure could happen here. And the cooler thing about that is when they go watch these corals spawning, I think Kelly and Ari are here, and they were watching them as well this year. When they go back and watch these juveniles spawn, they spawn like crazy. Like every mouth of the colony is spawning like crazy. That's really important because larvae grow, or settlers grow slow. These things are way smaller than the fragments that are used in outplanting. So you could, for a long time, say like larval propagation, what difference is that going to make? There's a multiplier here when we grow corals from eggs. They're really young and they're really fecund, and they're going to be doing a disproportionate job at making the next generation of larvae on their own. So major props to Secor for that vision and for getting those corals out there so that we could see only 4 and 5 and 6 and 7 years later what a difference that'll make for the population dynamics. That's super, super cool. I've been doing a little skunk works project on the side to try to understand how to help corals survive the settlement process. This is the slowest moving project in the world. I think I get like 4 experiments done a year. And I think I've had like 7 different student interns all work on this one step at a time. But starting like 9 or 10 years ago, we started settling corals on surfaces and growing the bacteria that live on those surfaces, and then screening the bacterial isolates one by one in little multi-well dishes to see which ones would trigger the larvae to go through settlement and metamorphosis. And then we would grow those as cell suspensions and wash the cells and then add them during settlement. And a couple of years ago, we got a result where we saw something like 7 times higher survival when we put them underwater for just a couple months. But it was a super, super tiny experiment, and there was no way I could say like, this is gonna work forever. This is like 70 corals. So my 2 awesome students right now, Daisy and Lucas, this year raised hundreds of Faviolata settlers, all replicated and interspersed at every step of the process. And last week they went and scored their most recent batch of settlers. 70% of the control settlers survived, and when we added these probiotic bacteria, we got 109% of them to survive. We are getting to a weird part of coral biology where the ARC code doesn't work anymore because we wouldn't expect any to come back alive. You find extra settlers if one survives and gets its symbiont, and then you find out it was actually 2 next to each other and you scored it as 1 at the beginning. So now we have the awesome problem of 109% survivorship and no idea how to do stats on that. That's awesome. Help, somebody help me. The beer people will meet over here, and then the stats people can come help me as well. Okay, and then our next breakthrough is the remix. Tom called me this year and said, we want Mary to bring cryopreserved sperm to Curacao and fertilize eggs in Curacao, and then we're gonna send them to Florida to get settled and grown by Carrie and Chris. And I said no, and then Mary said no, and then Mary called me and she goes, I said yes. I was like, okay, I guess we're doing this. So we had the great fortune and great adventure this year of having Mary and her team join us with a shipper of coral sperm from the USDA germplasm repository, try to collect sperm in Curacao, freeze that, take frozen coral sperm from Puerto Rico and frozen sperm from Florida, and then try to get any and all of that to fertilize eggs from Curacao. And the reason that that was significant in terms of well, there's 2 reasons that was important. One was we didn't know if corals in different populations get interbreed. So Curacao is the Eastern Caribbean, and Puerto Rico's in the middle of the genetic sort of transition zone, and Florida's in the Western Caribbean, and they're genetically isolated populations. And I was like, I don't know if these things can breed. Sperm-egg binding proteins evolve really fast, and I was like, I'm not sure this is gonna mix. And if they don't, we have 2 separate species to deal with and 2 separate conservation disasters to deal with. So trying it just to know was important. But then if you are also able to bring genetic diversity from one population to another, then you can play the remix game. You can start sorting through all of those combinations and looking for the ones that are gonna survive our human world. So the scene at the lab looked like this for a couple of months. Mary and her team did a ton of cryobiology training, and some of the steps are so delicate and nuanced and have to happen so fast that we actually practiced without samples. We just did like dry runs of the whole spawning night. Val Chamberlain and her team from Secor were amazing at accommodating the fact that we were all trying to catch a ton of material from a species that nobody could figure out this year. Nikki Fogarty said that this was the weirdest acropora year she's ever seen, and we had to dive 30 times to catch it. No big deal. On night 28, you're like, I think I might be going nuts, but I've been watching for 28 days, so I know they haven't spawned yet. So there were dribbles here and there, but we still hadn't seen a really massive spawn until the 29th and 30th days that we dove for proper spawning. And this is going to be the subject of Mary's talk tomorrow. We're dropping a preprint on this so that we can do some press and people don't think we're bullshitting. But just the short version of the story is it worked. There you go. Keep that a secret for like 21 more hours. That coral, those corals they live in Florida. Their mom is from Curacao and their dad is from Elbow Reef 2 years ago. That is nuts. That's totally crazy. And when Ileana was talking about sperm motility, I'm happy to report that the coral samples that we collected in Curacao had really good motility, both before freezing and after freezing. And so that's great, because that means it's gonna be an awesome place to do a bunch of gene banking and hopefully sort of work out even more methods for how to operationalize this. We've got 3 species in the gene bank at Carmabi now, and Mary's team was really strict in preaching about, like, you have to freeze these at 20 degrees Celsius per minute, because too fast or too slow, the physics doesn't work. They don't dehydrate the right way, they get ice crystals, they explode, they will die. So 20 degrees Celsius per minute was the thing we were aiming at for months. And then she just casually dropped this paper where she's like, also we can freeze and thaw coral larvae, and the way you heat them up is it's 4.5 million degrees Celsius per minute. It's like, okay. So it's a little bit different philosophy for the larvae compared to the sperm. Those are also larvae that are like coated in gold particles and they're heated up with a laser. So we thought we were doing some pretty high-tech stuff, but Mary is way far ahead of us in all of that. This is really cool. I think that I think they can do 12 at a time, but it's real. So to wrap this part up, this study system was about to go off, you guys. This was something that was really impossible until recently, and everybody who believed in it was definitely a little nuts and a little high on adrenaline. And now, thanks to the Coral Restoration Consortium and all of these geniuses in their factories all around the world, we're making this into a real study system. I almost can't believe that that's even real. I have 2 minutes for 3 unpopular opinions. Should be plenty of time. So instead of giving you a list of next generation technologies and list all the things we could do with larvae and all the things that we shouldn't do with larvae, I just want to plant the room with 3 ideas for how we might think about where we go next in coral restoration. It's not just about high temperature. I heard a funder at a conference once shit talk somebody's larvae work because they weren't heating up larvae in hot water and trying to get larvae to survive hotter temperatures. Naomi Oreskes studies the scientific consensus around climate change. These are thermal anomalies from the Western Hemisphere for the last 6 months. And one of the things she's really adamant about is that a lot of the world's gonna get colder, a lot of the world's gonna get drier, or wetter, or hotter, or more variable, or all of those things, but in different gears. So let's be really, really careful about what we think climate change is and what we imagine it is in our head, because there's gonna be just as many corals in weird temperature regimes as there are corals that are just getting straight baked. And what if we succeed? What if we grow all the corals to be better hot temperatures, and then DARPA changes climate change with some sulfur dioxide aerosols in the atmosphere, and then we have corals that grow in a temperature that doesn't exist here anymore? Let's not be so narrow-minded in the way we think about climate change that we miss the points that Ileana made about preserving diversity as a whole and getting these things ready for anything. Syrian wheat is being used to rescue the wheat of the Midwest right now from a pest. It's a little hotter, it's a little drier, but that's not the problem for the wheat. It's a problem because there's a pest that then eats the wheat that's kind of released from competition at those temperatures. So the wheat, the gene bank, the agricultural gene bank in Syria is now being used to breed genes into the wheat in the Midwest of the US to make it resistant to a pest. We have a lot of diseases and surprises and nasty biological things ahead of us, and we can't predict them. And really, this is our chance to save as much of the diversity as we can, so we can shuffle through all of those cards and try to come up with solutions to the stuff, the Donald Rumsfeld unknowns that we don't know, whatever it was. Okay, opinion 2. It's not just about gadgets. There are a lot of awesome funding sources coming online, including a lot of money from the tech world. And there is a tendency to then make the funding for tech stuff and gadgets and stuff. we need to be careful about a gadget-first approach for a couple of reasons. The biggest breakthroughs so far are the ones that people are making with like a tank of liquid nitrogen or an auger plate or a 10-gallon aquarium, and the growing and the husbandry doesn't need necessarily robots and satellites at every step of the way. If we also make our funding contingent on building gadgets, we're gonna magnify the inequality between rich countries and poor countries. If we make coral restoration something you can only get funded if you make gadgets and only something you can do if you own gadgets, we're doing ourselves all a disservice. So yes, let's go get all the money and all the contests and make sure all the money goes to corals and not any other kind of conservation. But let's also be careful about how much we fall in love with gadgets. This is a strawberry picking robot, and the reason that this robot's actually really bad at strawberry picking, but the reason that they need it is because they're really good at growing strawberries. So let's get really good at growing corals, and then we'll build our robots. Tom's roping me off the stage with a hook. We have a lot of natural history left to do, and if you gave me $100 million to fund what should happen next, most of it is gonna go to people who are making great progress and say, do the next crazy idea you have. And finally, we can screw up the scale up. We can do stuff too fast without understanding the principles at work and exhaust ourselves and set ourselves back a lot. Let's try to take the Six Sigma approach to every step of the process. Find something that you could do in that process and figure out a method that makes it work really reliably all the time. That's right now still a way more important contribution than trying to do something the most number of times possible. Once upon a time, teosinte was completely inedible, and now we can grow fields worth of it. We're doing the same thing, and we're making progress. We just have to keep in mind that we need time. Let's not panic. Recap, this works, Coral Restoration Consortium. We're about to figure out all the next steps, and this whole study system is really turning on. Let's not be too simple-minded about how we think about the future, and let's be smart when we scale up. We overestimate the change that happens in the next 2 years, and from what you've seen now, we clearly underestimate the change that's gonna happen in the next 3. So don't panic, guys, we've got this. And I thank you for your time, and I'll take your questions.
Questions for Dr. Marhaver. If you want to come up to the microphones in the 2 aisles, that would be great. And this time, I'm going to ask you all to just say your name and affiliation if you have a question. Please go ahead.
Hi, Sara Frias Torres. On behalf of Vulcan and the Paul G. Allen Family Foundation, we congratulate you and your team on the completion of the Assistant Gene Flow project.
Thank you, that's very sweet.
I have to say that I was in charge of analyzing all the ways the project could fail. And on the last line of my report, I said, but it might work. And that line convinced the people with the money to give away the money. But I wanted to ask you, could you explain a little bit, what was the secret sauce that made this project successful, and what will be the bottlenecks to replicate this same experiment somewhere else?
Oh gosh, okay, so thank you for those kind words, and also thank you for pointing out how improbable it was that would work. The secret to it working was to try to figure out every possible thing that could go wrong and try to plan for it. And then even still stuff went wrong and we had to adapt. So I was in charge of getting larvae into coolers to Florida. So I put temperature loggers in my luggage and I studied the temperature of the American Airlines cargo hold back and forth a couple of times. And then I tested different coolers in and out of the sun with a little temperature probe in them to figure out how many hours that much water could stay between 27 and 28 in that Caribbean sun. And I took a cropper and I put them in the cooler and I shook them and I drove them around town and I put them in and out of my air conditioning. I did all sorts of crazy stuff to try to understand how to be sure they would survive being transported. But then we started collecting little dribbles of spawn and the eggs were fertilizing themselves. Or the tubes of spawn were fertilizing themselves. So I didn't even anticipate that one of the huge hurdles would just be finding eggs that didn't already get fertilized. And it was probably because these stands of Acropora are so big, they're probably chimeras, and they probably have somatic mutations. But then we had to have a whole other strategy for how to get around the fact that they were getting fertilized 2 hours before we were trying to fertilize them. So we tried to anticipate every single thing. We spent a ton of time on the phone with each other just talking and planning and amping each other up, and then we just stayed nimble. And I think part of maybe the other thing that helped was the fact that we knew we had a baton and that somebody was waiting to take the baton, and this was this pressure to like not drop the baton and hand it to the next step. I think if you were doing it on your own, you'd be like, whatever, it didn't work, it's just my stuff. But to have the pressure of the group was a thing that helped also.
Thanks. Next question.
I'm trying to use up all the minutes so I don't get any hard questions.
I'm Ilsa Kuffner from the U.S. Geological Survey. Your observation that the colonies that are raised from larvae are more fecund than you see in the natural population on the reef, that is totally fascinating. And I'm wondering if, and that's new to me, I didn't know that.
That's Secor's news, and I'm just a messenger, so do not
Good job,
give me
Secor.
the credit for discovering that.
Okay.
I'm just proud of them.
So I'm also wondering, and you know, my thoughts are going back to Ileana's talk about the age of not only some of these genets, but of these colonies and being asexually reproducing for, it could be centuries. So I'm also wondering about whether colonies that are reared from larvae calcify faster or have different phenotypic expressions that we need to investigate. So it's really exciting to see that result. And so do you have any, my question is, do you have any hunch whether they do calcify faster?
Oh
Just
gosh, I
watching
don't
them
know
grow.
the answer to that, but if you find like Carrie O'Neill and Chris Page, they have been staring at these different populations of settlers for a few months now, and they'll probably have good anecdotes for you. Another thing that we found in Curacao recently was that corals in better looking, healthier reefs also seem to have more lipids in their tissues and make more babies. So there's probably the fact that if you're younger, you've got all your genes working, your immune system is better, you're growing fast. And there's probably also the fact that if you're in a healthier reef, you're able to take more, eat more, and use it for reproduction because you're not using it for defense and survival. For a long time, we would have just said, like, that would be great, but settlers grow so slow, it'll be really hard to ever figure out those studies. But now all the people who are doing settlement and grow out are getting so good at that they're able to get pretty decent sized polyps or colonies after 6 months, and then microfragment those. So we can answer questions like that in less than a 12-month period now, whereas before it would be basically undoable.
Well, I study calcification, and I have a network of calcification stations. I would love to test this. So,
Awesome.
Secor, somebody talk.
We'll find you
come
and we'll
find
find
me.
Chris and Carrie, and we'll
Because this could be
brainstorm a
easily
scheme.
tested. Cool.
Thanks,
All right,
Ilsa.
perfect. We've used our time and built some collaboration. Please join me in thanking Dr. Marhaver again.